Hi everyone, I am Thabang Nora Msimango. I am currently a PhD candidate at the University of Pretoria in the Department of Plant and Soil Sciences, as well as the Centre of Excellence for Food Security. I am working on a food safety quantitative microbial risk assessment project for my PhD, which is a build up onto my master's project, which I did by collecting fresh produce from schools around the Gauteng province as well as the Mpumalanga province. And after we collected the fresh produce, we tested it for potential foodborne pathogens that could be in the food and cause foodborne diseases in school-going children. In addition to the fresh produce, I also collected water samples, irrigation and tap water samples from the schools, soil samples, as well as kitchen surface swabs. And this was for us to try to determine what the school environment, what the impact the school environment and the hygiene at the school has on the safety of the food. And what we found was that most of the samples were of good quality and safe for consumption. However, there were a few samples where we found potential foodborne pathogens, and these foodborne pathogens were also resistant to antibiotics, which are commonly used when people have foodborne diseases. The results of the study indicates to us that food safety in schools needs to be considered very importantly, especially with regards to hygiene in the kitchens, to ensure that the food does not get contaminated, especially because with young children, they are at most risk of getting diarrheal diseases from foodborne pathogens. And foodborne diseases affect them a lot more than they would affect a normal healthy adult. And there's also the fact that they are immunocompromised children who, if they got sick, then they would their health would be compromised in comparison to a normal healthy child. So yeah, we went back to the schools and we shared the information with them. We also shared ways in which they could keep the food safe. Some of the things we shared with food handlers and teachers at the schools was to wash their hands properly with soap and warm water, and to also wipe surfaces in the kitchen with warm soapy water, and to also wash the fresh produce. This work was captured in my master's dissertation, and the work also contributed to a book that was on school feeding around the world, where different people from around the world were involved and shared their research on school feeding programs. And it was also included in a paper that was published in the Food Science and Nutrition Journal. The one question that I had after my master's was what is the actual risk to the children or to the consumer when they consume food that is contaminated with foodborne pathogens. And that led to my PhD project, where we use quantitative risk assessment to actually predict or estimate the risk that there is to the consumer if you eat food that is contaminated with bacteria. So at the end of my PhD, I will be able to say how many people could get affected from eating a type of food or fresh produce that is contaminated with this pathogen. And yeah, that is also important because it helps us determine the pressure on the public health system from foodborne diseases. But quantitative risk assessment also helps us determine the point in the supply chain where the risk is high, and what is nice is you can target those points to decrease the concentration of the pathogen, and by doing that you are decreasing the risk to the consumer.